So good morning, everyone. As Connie said, I'm Kristine Witt. Along with Stefan Fuhler, I co-chair the Genotoxicity Working Group. And today I'm going to provide you an overview of the activities and the progress that our group has made over the past year. Julie, next slide. Our current members are shown here. They come from Europe, Canada, and the US. And as Connie mentioned, they represent industry, academia, and government organizations. Next slide. Our mission is to develop a screening strategy that can reliably identify botanicals that have evidence of genotoxic potential with future applications for evaluation of associated human health risks. Julie? Our group is fortunate to have available to us a number of well-characterized and validated in vitro assays designed to assess the genotoxicity of individual chemicals. Our task then is to assess the suitability of these assays for analyzing the genotoxicity of botanical extracts, which you all realize are very complex mixtures, often containing 100 or more chemical constituents, some of which are identified, some of which are unknown. We initially used, I don't know if you can see my cursor or not. Since I'm not driving this, I can't use the laser. But anyway, we have initiated our investigations by running predictive modeling on the identified constituents for each of the 13 botanicals that we received for testing. The models that we used were for predicting potential for gene mutation and for chromosomal damage, because these are the two major categories of genotoxicity. The point of silico predictive modeling was to get an idea of what we might expect with the laboratory testing of the complete extract, and then to compare the results between that laboratory testing of the complete extract with the information we had on the individual identified constituents for each one of our botanicals. For our laboratory testing, we selected the bacterial reverse mutation, or AIMS assay, and the in vitro mammalian cell micronucleus assay. This investigates structural and numerical chromosome changes, and obviously the AIMS assay looks at gene mutation induction. These two assays constitute the standard battery used for genotoxicity testing. Both assays are guided by OECD guidelines, the conduct of both assays, and both are required by regulatory agencies for genotoxicity assessment. Now, at a later point, we added the Tox Tracker assay to our initial testing scheme to provide insights in mechanism of action. This particular assay has gained increasing use over the past decade, and there currently is a OECD guideline in preparation for this assay. Now, for follow-up assays, we have a number of assays available to us. Again, of interest to our group right now is a multiflow assay, because it can provide both confirmation of the results from the in vitro micronucleus assays, as well as information on mode of action. In other words, it can help classify a test article as either a clastogen or an antigen. Click, Julie. To date, we have included, back one, please. We have concluded the in silico testing. We've included all three of the initial GENOTOX assays. And we currently are in process for the multiflow assay. We expect that assay will be completed by the end of this year, perhaps early in 2026. Julie, next slide. Okay, now I'd like to present to you 4 case studies which illustrate the type of information that we've gotten to this point. We're using these to evaluate assays for their appropriateness for testing botanicals. So the first of the 4 case studies is Aristolochia. The aristolochic acids from Aristolochia induced genotoxicity and renal and liver cancer in humans. These are documented effects. Aristolochic acids have unique DNA signatures that are linked to cancer, which aids in exposure monitoring among populations. And Aristolochia tests positive in standard tests for genotoxicity. And based on this information, it was selected as a true positive case study for genotoxicity. The second of our 4 case studies is comfrey root. It contains pyrrolizidine alkaloids, which are linked to hepatotoxicity, genotoxicity, and carcinogenicity. PAs are metabolically activated to form pyrrole adducts, which damage DNA and proteins. And comfrey root, based on these factors, was selected as a true positive case for genotoxicity. Next slide. Asian ginseng root is our 3rd case study. Studies by the National Toxicology Program found no carcinogenic or genotoxic effects in rats and mice with ginseng. Ames test results were negative, and in fact, in several tests, ginseng was shown to reduce the genotoxicity of known mutagens. Based on these factors, it was selected as a true negative case study for genotoxicity. Milk thistle is our 4th case study. NTP studies found no carcinogenicity and only minimal chronic toxicity in rodent bioassays. However, genotoxicity results were inconsistent with mixed findings in various assays. Based on this information, it was selected as a non-carcinogen case study with undefined genotoxicity potential. Next slide. Now let's look at the various tests that were conducted, the 3 tests that were conducted with these 4 case studies, as well as all 13 of our botanical extracts. The first is the Ames test. This is a test for gene mutation. This test has been around for some 50 years, and it remains today the gold standard for testing for mutagenicity. The test is quite simple. In this particular test, the test article, or the botanical extract, or the vehicle control is combined with the bacterial culture in a test tube with or without S9. S9 is an abbreviation for induced male Sprague Dawley rat liver S9. This provides enzymes to provide a metabolic capacity to the bacterial strains. Since they don't have a liver, they can't metabolize these compounds in the same way that humans can, so we provide them an assist. This is incubated for 20 to 30 minutes, then the contents of the tubes are poured onto agarose media that lacks critical enzymes, critical amino acids, I'm sorry. Only bacteria that have been mutagenized to be able to manufacture their own amino acids can grow on these plates. These plates are incubated for 48 hours. They're removed from the incubator, and using a colony counter, an automatic colony counter, we count the number of mutant colonies on the plates. And test articles that significantly increase the number of mutant colonies are concluded to be mutagenic. Next slide. Here are the results with our 4 case studies. Aristolochia fengi was positive. Asian ginseng was negative. Comfrey induced what was called an equivocal response. This is a response that was definitely not negative, but the type of response was insufficient to support a call for mutagenicity. In other words, the magnitude of the response may have been below what we would hope to achieve with a true mutagen. There may have been a lack of dose response. There may have been lack of reproducibility among repeat trials. For whatever reason, we didn't feel the response supported a call for mutagenicity. Milk thistle was positive in the Ames test. Next slide. This is a slide that shows the principle of the in vitro mammalian cell micronucleus assay. This assay can be performed in any rapidly dividing mammalian cell type. And again, it's like the Ames assay, fairly similar. It's been around for decades. Here's a progenitor cell. If this progenitor cell comes in contact with a compound that can damage chromosomes, either an acentric fragment or perhaps loss of an entire chromosome during mitosis may occur. And this, during cell division, the acentric fragment or the lost chromosome fail to incorporate into either of the two daughter nuclei that are formed after cell division, but they do remain behind in the cytoplasm, and the genetic material forms its own small micronucleus, which is similar to the main nucleus except for size, and hence the name of the assay, the micronucleus assay. So this assay can detect both structural chromosomal damage or altered chromosome number. Julie? Here are the results of the in vitro micronucleus assay that was conducted using HEPA-RG cells. HEPA-RG cells are human liver carcinoma cells. Because they're derived from human liver, they have intrinsic metabolism, and therefore we do not need to use S9 when we're testing with HEPA-RG cells. Now, the bar graphs show the responses in this assay. You can see for Aristolochia, there clearly is a dose-related response in the incidence of micronucleated cells. Here for comfrey, there's also a clear dose response in frequency of micronucleated cells. For both milk thistle and Asian ginseng, there is no response, no increase over the dose range in the frequency of micronucleated cells. Julie, next slide. Adding those results to our table here, we see for Aristolochia positive, Asian ginseng negative, comfrey was positive, and milk thistle was negative. Next slide. We did a second in vitro micronucleus test, this one using human TK6 cells. Although these are human cells, they lack inherent metabolism capabilities, and therefore we did have to run this assay both with and without S9. I'm only showing here the results that were achieved in the presence of S9 so that we have a direct comparison with the HEPA-RG cells that do have intrinsic metabolic activation capabilities. So here we see the blue dots are the micronucleus response. Red shows cytotoxicity. That's an important factor to consider in these in vitro assays. But the blue dots here for Aristolochia clearly demonstrate an increase in micronucleated cells. For comfrey, in these TK6 cells, we do see an elevated dose point at this high dose. However, there's a lot of variability among the cell cultures here, and statistically, this particular dose was not seen as being different from those at the lower concentrations, and the test was concluded to be negative. The same situation was seen here with milk thistle, and the overall conclusion was that this particular assay was negative. For ginseng, there's no question that this is clearly a lack of response in the assay. Julie? So adding those results to our table, and I have put these in parentheses actually, because we're awaiting, these are preliminary conclusions, we're awaiting final data analysis and interpretation. We wanted to use a slightly different method of evaluating the data to make it more closely approximate the way the data is analyzed in the HEPA-RG cells. And we were supposed to have this information by this meeting, but due to the shutdown, it's been delayed. We should get it very soon, by the end of the year, surely. But for now, we'll go with what we've got from the preliminary analysis, and Aristolochia is positive, Asian ginseng negative, comfrey was negative in the TK6 cells, and milk thistle was also negative. Next slide. So providing an overall call for these 4 case studies, in other words, looking both at the responses for in mutation assays and in chromosome damage assays, we see overall we would call Aristolochia positive, Asian ginseng is negative, comfrey was positive due to the positive response in the HEPA-RG micronucleus assay, and milk thistle was positive based on the responses in the Ames test. These are different endpoints. They do not, the negative response in the HEPA-RG micronucleus does not in any way influence the fact that milk thistle gave a positive result in the NIEHS test, and therefore overall milk thistle is considered to be mutagenic. Next slide. Now here is a schematic showing the complexity of the tox tracker assay and the type of mechanistic information that it supplies. There are a variety of different endpoints in this assay, endpoints that look at DNA damage, aneugenicidity or changes in chromosome number, P53 activation. This is something that is seen with a number of different cellular stresses, including genotoxicity. Induction of oxidative stress. Oxidative stress can be a factor in DNA damage and protein damage. So a lot of information comes from this assay. The data graphs are very complex. Stefan Fuhler is an expert in this. He's not able to be with us here today, so I'm just going to provide the overall results that we got in the tox tracker assay. And Julie, can you show the next slide? So here we see that Aristolochia was positive in the tox tracker assay, Asian ginseng was negative, comfrey was positive, and milk thistle was positive. So note that tox tracker matches the overall calls that we got looking at the two assays, the AIMS and the gene mutation assays and the chromosome damage assays. Next slide. Now, as Connie mentioned, there's been an awful lot of outreach from all the groups. For our group, we've had recently 2 specific papers published, one by Zeiger et al. That was the result of a side project that grew out of a very detailed, comprehensive analysis of the massive NTP AIMS test database. And we thought our analysis would serve the gene tox community well. We recently published our screening strategy in food and chemical toxicology. We've had in a number of meeting forums, platforms and poster presentations at the 2025 Eurotox meeting, the Turkish Society of Toxicology, the Genetic Toxicology Association annual meeting that was held in Delaware this year, the German Environmental Mutagen Society meeting, the annual EMGS meeting. And I'll bring your attention to the fact that we have a symposium scheduled to be presented at the 2026 International Conference on Environmental Mutagens that will be held in Edinburgh, Scotland. The variety of audiences to which we have presented have provided us excellent feedback, generating high interest, good feedback to us, and a lot of encouragement. And so that's been well received by our group. Next slide. So we have a number of next steps. We will make all the data publicly available in SEBS. This is the database that's housed at the NIEHS. The data are there. We're just awaiting that final analysis for the TK6 micronucleus data, and then the data can be released to the public. We are in the process of drafting and considering how to publish our data papers. We have a massive amount of data. We can't put it all in a single paper, so we're discussing best ways to divide up the data to tell a good story. We would like to engage in discussions and collaborations with other working groups within the BSC. For example, we very much to engage in the discussion of ADME as well as chemical analysis. These would be very important to the interpretation of actual human risk down the road, and important to reconcile the in silico predictive modeling data for the constituents with the data derived from testing the entire extract. Future work proposals that we're currently discussing, these are all resource dependent. I was really thrilled to hear Connie say that we have got the MO, MO, Okay, I'm losing the name, but the memorandum of understanding approved again and funding restored for the next year at least. So hopefully we'll be able to do some of these things. We would like to run additional assays to obtain additional information on mode of action or for more direct application to human risk assessment. We would like to understand how best to integrate the in silico data into overall assessments of extract genotoxicity. And this goes back to what I was saying about the discussions with the ADME that's in hepatotoxicity and the chemical analysis working groups. We would like to be able to do lot-to-lot comparisons of certain key botanicals to compare the variability in the constituents and how that might affect test outcomes. And we very much would like to test additional botanicals to expand assay performance data. We have tested all 13. 13 is a nice number, but it is not a powerful number to be able to extend our conclusions to the whole world of botanicals. So maybe if we could test 20 or 25 botanicals for genotoxicity, we would have a more robust data set to better understand our performance data. But we are very pleased to date. We do believe that the tests that we have selected for evaluating the genotoxicity of botanical extracts will indeed be successful in identifying those that have genotoxic potential. And with that, last slide, Julie, I will thank you for your attention, and I'd be, if there's time.
Thanks, Kristine, so much. That was really excellent going through what's gone with GeneTox. So, what's new for an online talk? We're a few minutes off, but it was mostly my fault, Kristine, not yours. But I do think it's worth going through the questions because we have a little bit of buffer time at the end too. So one question that came in, Kristine, could you talk about how we selected concentrations for these assays?
The concentrations for the assays are based on cytotoxicity. These are all in vitro assays, so they're tested up to a point of cytotoxicity, and then multiple concentrations below that limit dose are tested so that we have a well-defined concentration response curve.
Yeah, and I'll just quickly add too that we did some of this based on limit of solubility and then adding on cytotox.
Absolutely. Thank you.
Yeah. No, thanks, Kristine, too. There's a similar question too about, how does this compare to what's in supplements? Do you want to talk if we did or didn't consider that?
In terms of the amount of the concentrations that we tested and how they
Yeah.
to the composition of a supplement, that's an excellent question. And that is something that gets into the area of human risk and extract composition, and also constituents in the extract. So in some cases, like aristolochic acid and comfrey, we know what the genotoxic constituents are. And we actually have a side project that's going on right now that is looking at the activity of the constituents in those two genotoxic extracts compared to the overall results of the complete extract. That will give us some information that could then be extrapolated into the amounts that are found in, well, aristolochic acid and comfrey, hopefully not either.
Hopefully not those.
Being taken, but that approach can be used to understand the relevance of the doses that we're testing to the human exposure situation. And that is definitely something that we want to look into down the road, and will probably require some additional testing to make that connection. That's an excellent question.
Thanks. So there's one from Kaylee Beans who asked about milk thistle, that basically that we got the one positive assay, excuse me, in the Ames, but it was negative in the other. So is that due to kind of a conservative approach that you would call it positive overall?
No, it's because it's a completely different endpoint. We're testing for both mutation induction, which is one category of genotoxicity, And we're looking at the capability of the extract for inducing chromosome damage. That's another category of genotoxicity. There are some genotoxic compounds that induce only gene mutation. There are some that only induce chromosome damage. There are some that induce both. And whether they induce only gene mutation or only chromosome damage does not mean that they are not genotoxic. They are genotoxic. That is the type of damage that they induce. Does that help?
No, that's great. Yeah, if there's any follow-up, I'll direct it to you, but I think that's really helpful.